Hi everyone, I'm Owusu presenting on aminoglycoside resistance, otherwise known as AGS. So, what are they? They are a group of natural or semi-synthetic antibiotics used in veterinary and medical sciences. Examples are gentamicin, neomycin, etc. Resistance mechanism. Resistance to aminoglycosides occur in three main pathways. One, enzymatic modification or inactivation, reduce permeability, and increase efflux. And the third one is modification of AGS target size. Enzyme modification. This is the most important and well studied mechanism for AGS resistance. Bacteria produce AGS modifying enzymes that modify the AGS antibiotic, inactivating and preventing it from binding to the ribosomes, cysteine S RNA. Enzymes are grouped into three subclasses. We have the AGS and acetyltransferase that cause acetylation. We also have AGS nucleotide transferase, which causes oxygen adoration. That's addition of adenosine monophosphate goes to oxygen atom. And the next one is AGS phosphorotransferase, which causes oxygen phosphorylation, adding phosphate group to the oxygen atom. These enzymes usually occur with other resistance on mobile genetic elements, leading to multiple resistance in pathogens. So the second route of resistance is reduce permeability and increase efflux. Here, modification of lipopolysaccharides and low energy bacterial population is responsible for the resistance. Occur by reducing the amount of antibiotic that gets to the bacterial target size. And this is achieved by A, reduce permeability, and two, efflux forms. And these are the bacteria transmembrane protection that actively pump the antibiotic out of the cell, hence making the antibiotic ineffective. Then we have the third mode of resistance mechanism called modification of the AGS target sites, where AGS normally binds to the cysteine S ribosomal RNA of the 30 S ribosomal subunit, which is essential for bacterial protein synthesis. The development of resistance occur here by modifying these binding sites, thereby reducing the ability of the antibiotics to bind or the affinity of the antibiotics. And that is, you also have mutations in the cysteine RNA gene, for mutations in the A site of the bacterial small ribosomal subunit, where edges lead to resistance by disrupting hydrogen bonds between RNA and the antibiotics. Why is this important? Why is the resistance important? One, Resistance to aminoglycosides are critical because these are very effective medicines used for treating multi-drug resistance infection, particularly gram-negative and gram, gram-negative bacteria. Resistance to this antibiotic therefore means losing one of the most important last resort options. Again, the gene encoding NAMs are often found on mobile genetic elements such as plasmids, allowing them to spread rapidly between different species of bacteria. And the next important is that AGS resistance worsens hospital infection outcomes by preventing effective therapies and reducing the utility of the aminoglycoside antibiotics. And the fourth is that the AGS resistance has become a growing public health concern, helping in spreading of infections in hospital and causing multi-drug resistant infections. How did it evolve? It didn't start just a day. Resistance to AGS evolved gradually under selective pressure from both natural ecosystems and human antibiotic use. Naturally, these are produced by streptomyces and microspora species, which are in the soil. And since these producers needed to protect themselves, they evolved a form of AGS modifying enzymes. And this actually started generating some resistance. Then we have genetic mobilization and acquisition, called horizontal gene transfer. Other bacteria in the environment acquire this resistance through horizontal gene transfer and were largely facilitated by the mobile genetic elements. Clinical use and selective pressure. Widespread usage of AGS has also created intense selective pressures where bacteria resistant genes survived and multiplied, spreading the traits to others. Bacteria develop resistance through enzymatic modification. And the fourth one is genetic spread. Resistant genes spread via plasmids, integrons, and transferons, especially in hospital environments. How is AGS resistance detected? So far, we've learned two of the methods, the Dix dilution or the Kirby bar method and broth and agar dilution method, where uh, an antibiotic impregnated disc is placed on agar needed, seeded with bacteria. The antibiotic diffuses, creating a concentration gradient. So to detect the small or absent of inhibition, that is area where bacteria don't grow, indicates resistance. And for the broth agar, bacteria grow in a series of tubes. And here, the detection is done by the lowest concentration with no visible growth is the MIC, as a minimum inhibition concentration. This value is then compared to clinical breakpoints or benchmarks to determine if the strain is susceptible, intermediate, or resistant. And we also have the gradient diffusion or the U test, among others. How is AGS resistance detected? Continuous. We have other testing protocols such as high levels AGS resistance used for serious enterococci infections. High level aminoglycol resistant testing is critical because there are some diseases that need very high concentration of drugs and also to detect the presence of resistance. Then we have genotypic detection, here where you have polymerase chain reactions, whole genome sequencing, among others. Therapeutic strategies for this resistance. One, optimizing aminoglycoside use. High dosage, extended uh, intensive interval dosing. Here we increase peak concentration to kill bacteria while reducing toxicity. Then we have therapeutic drug monitoring, where drugs are adjusted dose based on serum concentration, kidney function, and infection sites. Combination therapy, here the AGS antibiotics are often combined with other antibiotics to increase their efficacy against resistant strains. Therapeutic strategies continued. Use of next generation aminoglycoside. We have plasmazine, plasmazomycin, according to FDA 2018 approved. Semi-synthetic AGS designed to evade most of AGS modifying enzymes that causes resistance. I have alternative antibiotic. Here, when AGS fail due to resistance, carbapenems, etc., are used, and others like tigecycline are also used. Then we have the novel and experimental strategies, where the use of artisans, oligonucleotides, CRISPR, SRR, Cas-based, and pump inhibition, among others, are used. Other materials I found interesting in the book and through my research around the work. I found out that complex resistance mechanisms among bacteria. I also found out that there is a rapid spread via mobile growing elements, that resistant genes are frequently carried on plasmids and transposons, enabling fast transfer across bacterial population and species. And the fact that there's a new countermeasures being developed by researchers to actually control aminoglycoside resistance. And these are the references. Thank you very much.